Miss Davey, what role does cell division play in living organisms?
All right, so we looked at cells. We know that cells are made up of organelles and that these organelles will coordinate to maintain that stable internal environment, which we now know is
Homeostasis.
nice. But that cells are going to need to grow, right? So organisms get bigger or need to maybe reproduce. So that's where cell division comes into play. If a cell just literally gets bigger, the cell membrane can't control the amount of things going in and out enough efficiently so that the cell can maintain that homeostasis. So it's really important that a cell doesn't get too big. And we know that. Like when you were conceived, the sperm went into the egg, you were a zygote, which is a fertilized egg. You're not just getting a bigger cell as you get older. You're not a ball of cell, rolling around campus. So those cells are going to double and divide. Now, when they do that, that's called mitosis. It doubles its information, and then it'll divide into 2 cells. And then those 2 cells will double their information and divide into 4 cells. Why do you think we need to double the information?
So I assume we need to double the information so that when they divide, we don't lose any of that information. Is
right,
that correct?
Because all of your cells need that complete set of chromosomes. There's 46 chromosomes in all of the body cells of a human. So when we take a look at those chromosomes, it doesn't just like, okay, let me just double it and then divide into 2. There's an orderly fashion that it does that. And that nuclear division of that genetic information is called mitosis. So I've got a way that I'm going to help you remember mitosis.
I'm ready.
All right. Crazy names. There's interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. You will know them and love them.
Okay.
Interphase is that doubling. That's where the DNA is doubling. It's not really part of mitosis. It's just the first part of the cell cycle. So it's getting the cell ready to be able to actually divide its nuclear material. So when I teach this in class, we use our hands. So get your hands up. This is your interphase. It's just growing, getting bigger, doubling its information. Then we're going to go through the nuclear division because the nucleus is where the DNA is stored. So when that happens, the chromosomes, or all the DNA is going to coil up into those like X-shaped chromosomes that we know of, and it's going to start to prepare the cell for division. So we do prophase is the preparing. So interphase, growing, prophase, preparing. And it does that. It has these organelles called centrioles that will go to either side. They'll spit out these little spider web looking things called spindle fibers. Then the chromosomes are going to line up in the middle, right? So interphase growing, prophase is preparing, metaphase in the middle. And that's what you would see is those X's lined up into the middle, right? Then it needs to divide. So it's going to come apart in anaphase.
Anaphase.
Telophase is where we will now have a new nuclear membrane forming around the new sets of DNA. And then finally the cell splits in cytokinesis. All right, so you ready for this?
Yeah, let's go.
Interphase is it's growing. Prophase preparing.
Prophase.
Where does it go for metaphase?
It does this.
Middle, metaphase, middle.
Metaphase.
Anaphase apart. Telophase 2 new nuclei. Cytokinesis done.
Cool.
So that one cell now just became 2. That whole process is called the cell cycle. And it's broken up into interphase, mitosis, where they're coming apart, and then finally cytokinesis is where it would actually split. And it's a little different in plant and animal cells, which we'll learn as we're going through the unit.
Now, are there ever any errors that happen in cell division?
There are errors, and before we get to that, let me just say one thing. That is called mitosis. Now, that was making body cells, where all of your cells have all the information it would need, right?
Okay.
Now, that's making identical cells. When you were born, right, or when you're conceived, I should say, your cells, though, aren't just all identical cells. You're not like a ball of identical cells rolling around either. So our cells do something called differentiation. There's gene regulation that goes on so that like your toe cell doesn't need to know the information of what color your eyes are. So it turns off or on certain genes. And that's why our cells will differentiate. Those are all our body cells. There is one type of cell in our bodies, though, that doesn't need all the information because it only needs half. And that would be your sex cells. Because then when the sperm and egg come together, it will make the complete set of chromosomes. The process of making those sex cells is called meiosis. And it's similar to mitosis. You're going to hear the same words, interphase, prophase, metaphase, anaphase, telophase, but it goes through it 2 times. So it'll go through PMAT, it's called prophase, metaphase, anaphase, telophase, twice. So that's what we'll be looking at is the making of sex cells is a different process called meiosis. errors will occur, and if they happen in those sex cells, that's where it can be passed on to offspring. So sometimes maybe the chromosomes don't separate all the way, and now you may have an extra chromosome on one of the sets. For example, the 21st chromosome is Down syndrome, trisomy 21. It's because of an error in gametogenesis or meiosis. So we will be looking at karyotypes, is which when you take a picture of all the chromosomes and you kind of analyze and say, okay, is there anyone that has an extra or missing or pieces of one missing or added parts? And that's how you can diagnose chromosomal disorders.